paying attention to the digestive activity that's happening at each location. Okay? Forewarning, I'm on drugs this morning, so if you need to slow me down.
Do these drugs contain caffeine or?
1 Vicodin and 2 etc. Okay?
Yep, that's caffeine.
We're
I got
screwed.
headaches and stuff.
Oh, that's not good.
But just to let you know, if you need to slow me down, go ahead and do so. I won't take offense. You can tell Jeff to kick me. All right, so remember when we talked about digestive physiology or hydrolysis, that not only is the water molecule split, but in that case, it also splits our carbon-carbon bonds. We have carbon-carbon plus water, and we get this as a result. Remember I said that this would happen even if we did not have enzymes. Okay? So it's been a while, and I didn't really emphasize the function of enzymes when we did Bio 430, because that was in the chemistry chapter that I let you do your own review. But if we have an enzyme acting here, it does not become a component. No part of that enzyme becomes a part of the product. All right, so what it's doing is increasing the rate of the reaction, increasing the, disturbing the chemical bond, so it's easier for the electrons to shift in their spinning around the different atoms. So it increases the rate, because this would take place anyway, it would just take longer to occur. So we have various enzymes located in different parts of the body. They are going to react under, or be functional under different pHs, because that changes what hydrogens or hydroxyl groups are associated with the enzyme or denatured. And then we have different hormones that regulate motility and the release of enzymes. So that's the focus of today. There will be some material as well on how the information, how the products are taken up by the body. But that movement of amino acids or sugars is very similar, if not identical, to what we looked at with sympaports and antiports in the kidney. So that integral process there should not be too difficult. So we're going to do just an overview of what happens to carbohydrates and proteins and lipids, so that we don't do this each time we come to that area. So carbohydrates are long chains of sugars. How many of you in here have had nutrition? All right, you guys can get up here and lecture. And so we have a specific enzyme, amylase, that is going to take multiple sugar groups, and a circle here is going to represent a ring, a single monosaccharide ring, as well as we look at later, individual amino acids. So when we look at carbohydrates, or polysaccharides, is what we're going to start out with. This equals a simple sugar. But I'm sorry about my voice. I woke up with this low, sexy voice, so it'll take a while to get over it. So we start out with this polysaccharide, and only specific types of enzymes will work at it at this position, and that's amylase. So when we have sucrase and maltase and lactase, they will not work on the polysaccharide. Okay? They will, at least not efficiently, they will work best on disaccharides. So that first step, and remember a double arrow here means it's reversible. That first step, the enzyme is identified as amylase. We have two sites for amylase, the mouth in salivary amylase, and the pancreas for pancreatic amylase. Okay? So as you're collecting this information and organizing it for yourself, remember I told you there would be a table on the exam. And one is the site of secretion, and the other is site of action. So for salivary amylase, it's secreted by the salivary glands, but it doesn't work in the salivary glands. Those are accessory organs. Food doesn't move into the salivary glands. And so the site of action would be the mouth. A slight difference. A little bit more distinct when we talk about pancreatic amylase, where the organ of secretion is the pancreas, obviously, but the site of action is in the small intestine. Okay? So that's a distinction when you see that table. Then so this is our substrate and our product. Then our disaccharides. So this is what I call the intermediate step. It can take place in the mouth, to some insignificant degree in the stomach, but primarily most of this will take place in the small intestine. Right? Our final step, we utilize our disaccharides now. And our enzymes are specific to specific disaccharides. So sucrase, lactase, and maltase. So if you've had nutrition, I'm sure this is somewhere in some file in your brain. Okay, shouldn't take too much to bring it back up. So our substrates obviously are going to be specific for that. So we have sucrose, lactose, and maltose. Obviously they're disaccharides. So the product of each reaction is going to be two simple sugars. Glucose is always one of those simple sugars, right? So with sucrase, our product is going to be glucose and fructose. What's the byproduct for lactase?
Glucose and galactose.
Glucose and galactose. And maltase is
Glucose, glucose.
two glucoses. Okay? So when we talk about carbohydrate digestion, we're not going to go through all of these steps again. This is what we're referencing. So these are final products because these are simple sugars or monosaccharides. And utilizing facilitated diffusion, symport channels with sodium, as we saw in the kidney, we can move glucose across the plasma membrane barrier of the digestive tract. Okay, protein. So we talk about polypeptides instead of polysaccharides. We break those down. There is pepsin released in the stomach, but again, that's not the primary role of the stomach. We also have trypsin and chymotrypsin from the pancreas, and those are the intermediate step enzymes. And then the final step enzymes are made by the small intestine itself. Okay, so we have a similar diagram. All we have to do is change from starches to proteins. I get to just do this. You can recopy or change the words. I get to just erase this word. And put proteins. The ring now represents an amino acid. So instead of polysaccharide, we have polypeptides. And depending on where we are, our enzymes for the intermediate step will be pepsin, trypsin, chymotrypsin. This is what we're gonna cover as we go through the organs, so you don't have to worry about which one is coming from which location. And then we get oligo, which means a few versus many. Oligopeptides and dipeptides. So we may have 3 or 4 amino acids instead of just 2. Our final digestion, then we have oligopeptides or dipeptides. And the enzymes that break this down are going to cleave off an individual amino acid either at the nitrogenous end or the carboxyl end of our protein. Okay, so I have carboxypeptidase listed here as the enzyme, but there's also aminopeptidase. And then we have individual amino acids. So the overall view is very similar to carbohydrates as far as the individual types of molecules, and we've got sugars versus amino acids. But what's happening, you can see I use exactly the same images, is going to be very similar. Let me emphasize again that without intermediate digestion, the final step of digestion won't occur. So the enzymes of carboxypeptidase and aminopeptidase are so inefficient that essentially we would be unable to have sufficient substrate to break down enough amino acids to make it worthwhile. Most of these intermediate digestive enzymes, most of the intermediate digestion takes place with enzymes from the pancreas. So the pancreas is essential for life, not just because of insulin and glucagon, but our intestinal enzymes and our complete digestion cannot take place without those contents of the pancreas coming through the main pancreatic duct. All right, our last organic material here is fat. As you're well aware, there's a great variety of fats, cholesterol, and so on. We're just going to look at triglycerides. So triglycerides are made up of 2 types of molecules. I obviously don't, as I previously, I don't have the hydrogens on here. But we have the glycerol, which is a 3-carbon molecule, and we have fatty acids. Okay, this is what we're going to be working on in lab today. So with the whipping cream that I just gave Sherry, we're not making ice cream out of it. And I don't think you want to drink it with the purple powder that's in there anyway. So I'm going to erase the board if you want to take pictures. And kind of diagram this up. It's on the screen, but it'll slow me down if I write it out for you as well. And notice it kind of gets hidden in here. This COOH, that's the acid component. So when we talk about amino acids, it has that. That's the acid part of the amino acid. And so it's the acid part of our long chains of carbons that's interacting with the glycerol. If you're wondering where to put that. This does not have an intermediate step, all right? We just use lipase. We have 3 sources of lipase. There is salivary lipase, there is gastric lipase, and pancreatic lipase. I don't know why we bother with salivary lipase. It's really not, provides any functional amount, but it is present there. So this molecule, let me change the color. That's the glycerol, okay? And tomorrow, when we talk about metabolism, and we talk about the breakdown of glucose, and we form glyceraldehyde 3, whatever, you'll notice that same 3-carbon sequence. All right? What does that tell you about where we can, if we don't have carbs, we don't have glucose or fructose, we can bring lipids into our glucose metabolic sequence at that location. So we can bring the glycerol components into our metabolic sequence where it matches up with these 3 carbons. Then we have our fatty acids attached. Again, I don't have the carbons on here.
Hydrogens.
Sorry, thank you. Hydrogens. All right, so when we break this down with lipase, we typically don't have just straight glycerol. It's usually a monoglycerol, although I wrote glycerol there, so we usually have a carbon or glycerol molecule with at least 1 fatty acid still attached. And then we have 3 fatty acids. So keep this in mind. If our substrate in our digestive hydrolysis lab today is going to be fatty acids, without drawing the diagram out for you, what would you expect? Our cream, what Sherry's doing it with right now is adding litmus powder to it, and it takes a while to shake it. And not all of it gets dissolved in there, so that's why she needed it so early. And I should have given it to her yesterday. But we start out with a purple lavender color. What pH does that indicate?
High pH.
Higher alkaline pH. If our enzyme is pancreatic lipase, why would that be significant?
Because you're going to create free fatty acids and lower pH.
Well, what's the pH in which our pancreatic lipase needs to work?
8 to 9.
Around 8 to 9. So I know we haven't gotten to that yet, but I'm just kind of trying to perk your ears up so when we do, it makes an impression. The bicarbonate ions from the pancreas are going to not just neutralize the acidic pH from the chyme of the stomach, but elevate it a little bit higher even to a pH of 8 to 9. So we don't want to start out with an acidic pH in our cream. The gastric lipase may work at that, but our pancreatic lipase will not. Okay? However, if our digestive hydrolysis experiment is successful, what color would you expect it to become?
Red?
Reddish to mauve, depending on how long it goes. It'll be pink to a kind of a reddish purple because of the release of fatty acids is going to provide free hydrogen ions sufficient to lower the pH of our product. Okay? Obviously, if we lower it a lot, our pancreatic lipase is going to stop working or not work as efficiently. We'd have to keep, adding additional. But that's showing that fatty acids are released. Okay, so it's one of the, not to answer your lab questions for you, but I am. That's one of the mechanisms or signs that we will have that digestion is occurring. All right, so starting with the mouth, may I erase the board? You guys have all this on the screen anyway. So mouth, due to salivary glands, has a pH of averaging around 6.8. So slightly below neutral. Obviously, we've got bacteria in the mouth, and so that can affect the pH as well. But when we look at what's occurring in the mouth, primarily what we have is salivary amylase, and salivary lipase. We don't have enzymes for protein digestion. Depending on how long you chew your food, right, it's going to affect how much time those enzymes have to be active. Would there be any absorption of final products?
No.
No. So unless you ingest simple sugars, right, or unless you ingest single amino acids, you're not going to have a product that is small enough to cross the plasma membrane. So you're merely preparing, again, the mouth is primarily functional for mechanical digestion. As we move down the esophagus, for the few seconds that it takes for peristalsis down the esophagus, there are no additional enzymes that the esophagus provides. Right? So for that period of time that it takes to move food through the esophagus, these enzymes will continue to function. But our submucosal glands are releasing mucus, not digestive enzymes. So then we get to the stomach. And that's a cool picture. Remember I talked about walking along the surface of the stomach? What would these openings be for?
Those are the pits?
Good. I was hoping I wouldn't hear gastric glands. Gastric glands would be deeper, but not as wide an opening. So these are gastric pits. What is this spidery web stuff that you see here?
Mucus?
The mucus. Remember the gastric pits and the top of the glands are lined with mucus cells. Not goblet cells, but mucus cells, for the protection of these cells against the acidity and the digestive enzymes. So looking at our components, what are the cells that secrete hydrochloric acid?
Primary?
Starts with a P, but not primary.
Right.
Parietal. I don't know why we like the name parietal, parietal peritoneum, parietal flora, parietal cells. So these secrete hydrochloric acid, which almost everybody talks about. Its role is to activate pepsinogen. Let me move this over slightly. To pepsin. However, at least in human stomachs, that intermediate step of breaking polypeptides down to oligopeptides and dipeptides down the stomach is really insignificant, okay? So as I indicated on Tuesday, I think I might have said something on Tuesday, in my mind, the hydrochloric acidity is more important in killing off the bacteria and viruses and pathogens that might be present in the food, because we don't really get much of this digestion occurring with the pepsin. So carbohydrate digestion. Our cells that secrete enzymes are chief cells. They're going to be making the pepsinogen, which is activated by hydrochloric acid to pepsin. They also make gastric lipase. No enzymes for carbohydrate digestion. All right, so you can nix that. Not only does the stomach not make enzymes for carbohydrate digestion, but it denatures by the acidity the salivary amylase that has come down from the mouth. So we block that. So, and there's no additional enzymes that are added to replace that. So if there's any digestion that occurs, it would be a small amount of intermediate steps for protein and a small amount of lipid. More importantly, the stomach is doing the mechanical digestion. So for the lipid, it's creating small lipid droplets to increase surface area, all right? So not a lot of even lipid digestion. The purpose is primarily for those components of the mechanical digestion. Absorption. Do we have any products of protein digestion that are single amino acids?
No.
No. Lipase only requires one step, but again, there's insufficient amount of the lipase, and really the emulsification process is ongoing. So there might be a tiny amount of fatty acids that are absorbed, but again, insignificant. Water is really not absorbed here, so again, drugs, okay? Alcohol or drugs that are lipid soluble. So some drugs are released and are taken in time-release capsules. So some of those might be digested by the enzymes, the acidity of the stomach, and be absorbable by the lipid soluble, be absorbable, is that a word? Absorbed across the plasma membrane. But other than that, we really don't have any significant absorption. Now, because these words are so small, this chart looks a bit off-putting, okay? So let's look at what it contains within it. These are phases of digestive processes, movement, and enzyme release. Our first motility, our first hint of motility and chemical digestion really takes place in the stomach and then progresses through to the small intestine. So it occurs in 3 regions and 3 sequences. So cephalic refers to head. And so just thinking about food, when I drive home, I go by a bakery. So if it happens to be the right time of day and I have my windows open and I can smell the food, the bread breaking, all right? Or I get home and my daughter's made chocolate chip cookies, that kind of thing, you start to secrete saliva, all right? So that's stimulation via taste and smell. You don't have to actually have food in the stomach for that to be triggered. Vagus nerve is our parasympathetic component that's in charge of esophagus and stomach and small intestine. So enzyme secretion due to nerve activity, motility, the peristalsis, contraction of our circular and longitudinal muscle is going to be under vagus nerve activity, right, as a stimulation. And that occurs without the presence. Just thinking about food or smelling or tasting can cause that to happen. Then we swallow food, or our body thinks we do, and the presence of food in the stomach, especially as we start to stretch it, is going to activate the vagus nerve and the local enteric nervous system to cause the same thing to happen. So we're going to see secretion of hydrochloric acid. We're going to see secretion of pepsin and gastric lipase from the chief cells. Right? That's happening in the stomach due to sensory stimuli in the stomach. So that's why it's called the gastric phase, because the origin of the stimulus is in the gastric phase. Okay? Now, in addition to the food being present and the nerves being activated, remember our enteroendocrine cells that release gastrin? So we have G cells, or gastrin release cells, releasing a hormone. Okay? Remember, I want you to recognize which of these chemicals are hormones and which are enzymes. Typically, our enzymes end in an ASE. So if it does end in ASE, it is an enzyme. All right? Pepsin and trypsin, however, end in an IN. Those are still enzymes. So you need to make an effort to keep that clear. If it's an ASE, amylase, carboxypeptidase, it's going to be an enzyme. But the pepsin, the trypsin, the chymotrypsin are still enzymes. You can't go completely by that ending. Gastrin, however, is a hormone. And it's an interesting hormone in that it goes into the blood from the capillaries in the lamina propria of the stomach mucosa and recycles back to receptors that are in the stomach. Okay? So not necessarily a paracrine or autocrine where it just diffuses a short distance. Because it gets in the blood, it's going to muscle, so it can't do a paracrine, autocrine